this by a very well-known person and a very sweet person like you. I'm indeed honored. At the outset, I would like to congratulate Dr. Vasundhara and Dr. Sinja for this initiative. And I'm indeed truly privileged to be the opening batsman of this entire program. And I wish you both very good luck in conducting this or in continuing this program. So with this, we'll start today's talk. It's a master class. So when we say a master class, it has to be kind of, one second. So we have to have the information in detail. I have tried my best to incorporate the basics as well as the, information of clinical relevance. So the scope of this presentation is we would start with the steps involved in fertilization. I would like to overview a few major events in vivo and in vitro fertilization as with special information on sperm capacitation and acrosome reaction. We would also look at the analysis of fertilization and then what are the different types of abnormalities we see during and after fertilizations. And since Dr. Vasundhara had mentioned it to me that like many of the clinicians may not know what are the factors which are actually involved in the success of fertilization. So I have incorporated oocyte factors because, like with sperm, we are very familiar. We know that the quality of sperm is very important. So I have kind of underplayed the role of sperm and I have tried to incorporate the role of oocyte in fertilization. And I have also included information on the interventions. What can we do? Like, can we what does the literature say? If we can do something on the stimulation protocols to improve fertilization, and then about artificial oocyte activation. Finally, summary and conclusion. Just want to turn on the timer. So we all know that fertilization is the union of two haploid gametes, the female oocyte and the male sperm, and which actually fuse to form a diploid zygote to initiate the prenatal development. Now, what are the major events in fertilization? First, the sperm has to navigate through the expanded cumulus cells of the oocyte. The picture is here. You can just look at the, the numbers. This is the first step. Second step would be sperm has to be recognized by the zona pellucida, the outer cover of the egg. And this actually would depend upon four zona pellucida proteins, that is ZP1, ZP2, ZP3, and ZP4. And some linked oligosaccharides are also involved. So basically what happens, sperm binds to ZP3, it undergoes acrosome reaction and binds to ZP2 and becomes competent to enter through the zona pellucida. Step 3 would be, like once the sperm enters, it has to fuse with the egg. And here the sperm binds to the oolemma, and the area where it binds is, there are small microvilli on the surface of the oolemma. So it just attaches to the microvilli, and a lot of associated membrane proteins are involved, and it forms a fusion pore. And it's just not enough the sperm enters the egg. There is also a process called as activation of the oocytes because, the oocyte, the chromosome and the oocyte as such has to get activated, should be ready for this union. And a lot of signaling cascade happens and there is cortical granule reaction. We all know that the cortical granule just accumulates on the periphery of the oocyte. And this actually is for blocking polyspermy through the modification of ZP2 and ZP3. Then the second meiosis is completed and polar body 2 gets extruded. Now, step 5, sperm contents have to be released in the ooplasm, especially the nuclear contents of the sperm. And the last step would be the formation of pronuclei, and the pronuclei has to migrate, like they're formed at different places, but they should migrate close to each other and be ready for the syngamy. Now, what just happens in the natural circumstances? This is a very quick recap. I'm not going to spend much time on this here. So what happens here is, after coitus, human sperm, which is actually deposited into the anterior vagina, it just moves up through the cervix because, the vaginal pH is acidic and there is a lot of immunological factors there, immunological triggers there. So to escape from that, the sperm quickly moves on, navigating through the cervical mucus, and it enters the cervix. Now here, the first step of filtration happens. Cervical mucus actually removes sperm with poor morphology and motility, and only a minority of an ejaculated sperm would actually reach the cervix. So when we start with million of sperms, at this step, we have, like many of them getting filtered there and the good ones moving upward. Now they start their journey in the uterus, and this journey is enabled by muscular contractions of the uterus. And what happens, a few thousand sperm actually would reach the uterotubal junctions. Okay. So here, the picture which shows you the uterotubal junction, this is the uterus. Okay. And this is the isthmus part of the fallopian tube. And you have the ampullary part of the fallopian tube here where the egg is there. Now, this picture is from a mouse. So you see multiple eggs in here. And I do not know if you can appreciate the sperm here, like a lot of sperm moving on in this region. And if you can just observe it clearly, you can see few oocytes in here, okay? And this is a fluorescent labeled picture where you can see sperm moving up in the through the uterotubal junction to the isthmus region. Now, what happens is when they reach the isthmus region of the tube, the sperm actually, like it stays in that microenvironment. And here a unique thing happens, there is a delay in the capacitation of the sperm, meaning they are stabilized, they are not ready for fertilization, they are maintained in that phase. And this actually can be, extend up to 24 hours. In humans, that is what they have found out. Now, when there is ovulation, when there is a trigger for ovulation, the sperm actually gets the signal and it becomes capacitated and hyperactivated, and it starts moving towards the tubal ampulla. Now, this process happens by a combination of change in temperature, that is thermotaxis, and also chemotaxis, where chemical components are being involved. Now, let us just look at capacitation. We did mention about capacitation, where the sperm is kept in a stage before capacitation. So what actually happens during this process? This is a physiological phenomenon which involves both biophysical and biochemical processes by which the sperm actually becomes competent to fertilize the egg. Now, this capacitation actually happens in a biphasic manner. Okay, it happens immediately after ejaculation. The initiation of capacitation happens in the female reproductive tract. And remember, this is required only by the mammalian sperm. So the first phase is during its transport through the cervix and the uterus. What happens is cholesterol and the other sterols which are present on the surface of the sperm are removed, and then there are non-covalently attached glycoproteins are also removed from which actually the sperm acquires it through its journey in the epididymis, are all removed from the surface. So this happens before the sperm reaches the isthmus region. Now, isthmus region, it just stays like that, but then when there is a stimulus there, the second phase of capacitation happens. Here, we see a rapid change in the biochemistry or the biochemical parameters on the sperm, where there is an increase in the reactive oxygen species generation. There is an increase in cyclic AMP. Cyclic AMP, we all know that it is very much involved in the energy process. And calcium levels are changed, and there is also change in tyrosine phosphorylation. Now, in response to these signals, sperm actually begins to express hyperactivated motility. And this is a very interesting phenomenon. If you just look at the image of the hyperactivated sperm, it has different kinds of movement. Okay, there is a flagellar movement. This flagella actually moves very vigorously, and there is a movement in the shape of 8. There is also some cyclical movement. So the sperm actually, does all these acrobatics so that it can just go through the cumulus cell and go through the zona pellucida and enter the oocyte. Now, these also elicit certain dynamic remodeling of the sperm surface. And here what happens is there are changes in the acrosomal region. And so that is also very important for the fertilization to happen. Now, so this is something which happens in vivo. What happens in vitro? Because, like we are all embryologists and reproductive biology specialists here involved in a lot of ART procedures. So how do we ensure capacitation in an in vitro condition? So first, we prepare sperm through a swim-up method and density gradient centrifugation. And the first step is removal of sperm from the seminal plasma. Now, seminal plasma has decapacitation factors. So we are removing that, and we are, providing a condition where the sperm actually can undergo capacitation. The second process is where the culture media components, the bicarbonate present in culture media, calcium present in culture media, can trigger capacitation, and then incubation temperature also can induce capacitation. So this is precisely the reason why they say that when you are collecting sperm for an ART procedure, it should not be kept at 37 degrees Celsius for a very long period of time. And then it should also not be kept along with the seminal plasma for a very long period of time because, like you induce capacitation. Okay, so which we do not want. We want the capacitation to happen just before fertilization. So as I said, so capacitated sperm, it moves towards the oocyte to reach the zona pellucida, and it has to start binding to the zona, right? And for this, the sperm has to, as I had mentioned earlier, there is remodeling happening in the sperm structure, and that remodeling is the acrosome reaction. Now, what is an acrosome reaction? Acrosome by itself is it's a Golgi-derived exocytotic organelle that actually covers the tip of the sperm. 40 to 70% of the sperm head is covered by acrosome. And this acrosomal exocytosis or the acrosomal reaction, it can happen only if the sperm is capacitated. And as I had mentioned earlier, it's a prerequisite for sperm to fuse with the egg. And it occurs at the zona pellucida after binding of the sperm. So what happens here is, first the outer sticky membrane, that is the plasma membrane, it starts to shed. It's very sticky. So, it doesn't shed off completely from the sperm, but it goes behind the head of the sperm and it just remains stuck to the sperm head. Now, the inner and the outer acrosomal membrane, if you can appreciate it here in the picture, they start to fuse, and then the content of the acrosome, that the hydrolytic enzymes are actually released. Okay, so that, and what happens is the acrosomal contents, when the hyaluronidase and then acrosin, when they are released, now the equatorial segment of the sperm, the mid piece here, this gets exposed. And this is very, very important for fertilization because the sperm binding to the oocyte happens at the equatorial region here. Now, remember, if a high proportion of sperm undergoes acrosome reaction prematurely, or there is an acrosome reaction deficiency in the sperm, both of them are correlated with decreasing fertilizing potential of the sperm in vitro conditions. Now, how does this acrosome reaction happen? Like we do not have these kind of stimulants, like the stimulants which are present in the uterotubal junction in the in vitro conditions. And it has been found that when you immobilize the sperm, that induces acrosome reaction. And that is precisely the reason why they say that, like, your immobilization has to be proper. And especially if you are immobilizing a sperm just below the neck region, like they have observed localized vesiculation. And in some cases, there is a complete disruption of the acrosome or complete loss of the acrosome. Now, this immobilization, you might be afraid that if I'm going to damage the DNA, the chromatin of the sperm, no changes in the chromatin were observed. And there is also another point which is actually found in the literature, that in assisted conception treatment cycles, okay, wherever we are preparing sperm for intrauterine insemination and conventional IVF, the incubation interval after sperm processing and the insemination should not exceed 3 hours. Okay, so this has to be kept in mind, because if you have a very long post-processing incubation, that actually can increase the proportion of sperm that undergo untimely acrosome reaction, and that actually can impact on your fertilization outcome. Now, so acrosome reaction has happened, the equatorial segment is exposed, the sperm actually, like enters the zona pellucida, and the next thing what happens is there is a change in the oocyte's membrane potential, because, see, these are all biochemical. It's just not a single phenomenon here. You have a lot of activities going on simultaneously in the creation of this life. Okay, so there is changes in the oocyte membrane potential. Then oocyte activation happens along with calcium flux. When we say calcium flux, it is just not one single incidence of increase in calcium. There is actually a cycle, a rhythmic cycle which goes on, where the calcium increases and it just goes down. And as I said earlier, there is completion of metaphase 2. So polar body 2 gets extruded. Then cortical granule exocytosis happens. Sperm head actually gets detached and it gets decondensed. See, many of the like people, when they are talking about immobilization of sperm, I have heard them saying cutting the sperm head from the tail. Actually, when we immobilize sperm for ICSI, we never cut it off. Tail has to be there. In fact, it is shown that without the tail, if you just inject the head alone, you will not get fertilization. You will get very poor fertilization. So sperm tail has to be there. And that the head gets detached inside the ooplasm. Now, the sperm protamines are replaced with histones from the oocyte. Sperm centriole gets deposited. We all know that centriole is the only organelle, apart from nucleus, which is inherited from the sperm. And then oocyte chromosomes actually progresses from metaphase to interphase. Then there is formation of the male and the female pronuclei. Now, movement and fusion of the male and female pronuclei, it happens because of the centrosome. And the sperm centrosome, it replicates, it splits, and it organizes a bipolar array that forms the mitotic spindle, which actually ensures that proper cleavage happens post this process. Sindhu, do you want to do the Q&A now? The first
Yes, ma'am. The basics we can do now.
Yeah.
Yes, ma'am.
So should I stop sharing my slide?
Yes, ma'am. Maybe you can stop sharing for a couple of minutes.
Okay.
Okay. So I hope everyone's sincerely attending, because I think it is very interesting the way ma'am is taking it forward. It's like listening to a story, actually. So I'll share a quiz now. I request all of you to participate. Don't worry about right and wrong. These are just 5 basic questions about what ma'am has just presented, just to keep you all engaged. So I hope all of you will participate. Okay, I'm launching it. All of you can try to attempt. Okay. Are you able to see it? I'm seeing a good participation. 30% have responded within a minute. That's really nice. I think we can take a maximum of 5 minutes, ma'am.
Okay.
Almost 2 minutes is done, and 65% have responded. Meanwhile, if any of your friends are trying to join and they're unable to, kindly ask them to join the YouTube link I've shared in the chat box. There is some small technical glitch that will be sorted shortly. I think we've reached a maximum number of participants.
Not getting quiz on the screen, ma'am.
Ma'am, I'm sorry.
Quiz is not appear on the screen.
Oh, it's not coming on the screen. Okay, I'll
Yeah,
just
yeah.
1 second. 1 second. Are you able to see the QR code?
No, ma'am.
Okay, 1 second. I've sent one thing. If you just click on that, one QR code will come. Through that also you can try. And in the chat box, can you please check? You have to click on it. If you click on it, it will open the QR code and you can scan it on your phone. I think the others are able to do because almost 80% have attempted the questions. Okay. So 5 minutes, ma'am. Shall we end it? Shall we end the
Yes.
poll?
Yes.
Yes. Okay, I'm ending the poll. I'm going to share the results now. Are you able to see, ma'am?
Yes.
Do you want to briefly discuss them, ma'am, with the right answer?
No, you can do it.
Okay. Okay. So the question is, where does human fertilization typically occur? 95% have gotten it right. It's yes, it is in the fallopian tube.
Sindhu, we are just seeing the question, Sindhu. We are not seeing the poll or the right answers shaded. It is just like a questionnaire. We are not able to see
Okay.
the results. Yeah.
Can you see the green tick selected, ma'am, in fallopian tube? Or
No.
okay. Okay, I'll tell the correct answer is written below, no, ma'am? Correct
No,
answer is coming.
no.
Are the others? Sujata, ma'am, are you able to see it?
No.
Okay. Can you just give me 1 minute to try this?
Yes.
Yes.
How about now?
We are
No.
seeing only the questions and the.
Yeah, we're not seeing the answers, Sindhu.
Okay.
It doesn't matter. I think whatever is there on your screen, you can just read it.
Read, yeah.
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 Okay, okay. I found it. I'm
Yeah,
sorry,
now
I'm a
it's
beginner
now
in this.
it's there.
Now you
It's
can
there.
see.
Yeah.
Okay. So where does human fertilization typically occur? 95% have answered it right. It's in the fallopian tube. How many chromosomes does a fertilized human egg contain? So that's a zygote. So we know that's 23 from sperm and 23 from oocyte. So the correct answer is 46. So 77% have gotten it right. It's not 46 pairs. It's 46. It's 23 pairs. So the answer is 46. Which part of the sperm cell contains enzymes to penetrate the egg during fertilization? I think ma'am explained in detail about how the acrosome comes and the acrosome and all those things. So the correct answer is acrosome. So majority of you have gotten that right. And I think the head is also not a wrong answer because acrosome is in the head. So to be more precise, it's acrosome. And what prevents more than one sperm from entering the egg? That is the zona reaction. So basically because of the zona hardening, multiple sperms don't enter. And the process by which sperm cells become capable of fertilizing an egg after entering the female reproductive tract is known as. So 70% of you have told it right. It is the capacitation reaction. I'm stopping. Yes,
Good
ma'am.
response.
So, ma'am, Sujata, ma'am, it's a success. Majority have gotten it right.
Yeah, good response.
Yes, ma'am. So, ma'am, I think you can start sharing and.
Okay. 1 second. Just a second. Okay, is it visible? Is it visible?
Yes, ma'am.
Yes,
Yes, ma'am.
ma'am.
Yes,
Yes,
ma'am.
Yes, ma'am.
Okay, okay, okay. So let's move on to the next, what to say, part of fertilization. Okay, so we kind of went through what is the basics of fertilization, what is exactly important, and in vivo so many things happens, but in vitro what is important and for fertilization to happen. Now it's about the analysis of fertilization. Now, see, this is something which I always emphasize, that embryo evaluation, okay, let it be fertilization, let it be cleavage assessment, or blastocyst formation on day 5, day 6, it should follow a standard protocol and timing. It should not be like a random thing. So, I just came by the lab, I had an early pickup, so I'm just checking this fertilization for the previous day, and it is just 15 to 16 hours. The next day I'm coming in a bit late. So, like I'm just checking it after 21 hours. It shouldn't be like that. There should be a standard protocol as per your SOP, and there should also be a standard timing. Now, this timing of assessment, for convenience sake, should be with reference to the HCG trigger or the insemination. Whatever time you do the insemination, so many hours post-insemination or so many hours post-HCG trigger, that should be your reference point. Now, see, another important thing is the maximum information you can get from this assessment would be beneficial. But at the same time, you also should remember that you should not expose the zygotes or even the embryos for a very long time to the ambient conditions. The culture condition, the optimal culture condition has to be adhered to. Now, so the balance has to be there. And definitely this comes with experience that, like when you are about to check something, for example, if it's a fertilization check, there are few points which actually it should be there in your mind. And then you look for that particular point in the zygote. So this would help you to do things very quickly. And it will also help you to look at things very, very carefully and in a very detailed manner. Okay. And then, so for this, like before you even do all these things, it is important to have an idea about the time points at which the most significant change can happen in the inseminated oocytes. See, most of this information has come from the time-lapse studies. Okay. So the first event, which we can see under the microscope, is the extrusion of the second polar body. Okay. So the moment you finish ICSI, within 1.5, 1.5 to 3 hours, you can just start to see the polar body getting extruded. Now, male and the female pronuclei, it forms from 6 to 7 hours post-insemination. You can see them faint, like in case if you are checking it early, you can actually see very faint pronuclei as early as 6 to 7 hours post-insemination. Now, these pronuclei, they come together somewhere between 8 to 9 hours. And after that, the halo actually forms. Now, halo disappears between 18 to 20 hours post-insemination. Pronuclear membrane breaks down at around 24 hours, and the first cleavage actually starts 3 hours later. So, like in one of our programs, we were actually, like routinely doing early cleavage check. So many of this information actually we got by observation, that when does this pronucleate disappear, and, like when the first cleavage starts. So, all these things actually we have observed it, and we have also seen these phenomenon under the time-lapse microscopy. Now, what are the markers of fertilization? We know that the zygote should have 2 pronuclei, and it also should have 2 polar bodies. And this is 16 to 18 hours post-insemination. Or if you're looking at the HCG trigger, then it should be 56 to 58 hours post-HCG trigger. Now, what do we expect at this particular time? So these pronuclei actually should be aligned and juxtaposed to each other. See, like this, they should be lying close to each other. Suppose if you are not, the pronuclei is not aligned, can you see a small, space between the pronuclei? This is most likely because of the sperm centriolar defects. And another thing which we have to see is pronuclei generally should be present in the center of the oocyte. But if it is not in the center, because sometimes the oocyte shape actually varies, right? Like it may not be a perfect circle. You can have elongated oocyte also, or with different shapes. In that case, just see if the pronuclei is present in the half which contains the polar body. So it should be somewhere, suppose if you divide this oocyte into 2 halves, your pronuclei actually should be lying in the half which actually is closer to the first polar body. Now, there is also another thing here. See, suppose if you are, this is the pronuclei, right? Like if you draw a line through the pronuclei, this axis, and if you are drawing a line through the polar body, now there should be a perpendicular alignment instead of a parallel alignment. Okay, this perpendicular alignment, it reflects the normalcy of the meridional plane, because what happens, subsequent cleavage divisions, when it happens, the genetic material should get equally distributed between the 2 daughter cells. Suppose if it is, say for example, if the alignment is kind of parallel, then what happens when the cell divides, one cell will get both nuclei, whereas the other cell will not get anything. And this cleavage, it will not go further. The embryos can get arrested. So this is something which you can observe. And there is also a finding that, see, suppose if you're drawing this line and if you're looking at this angle should be less than 50 degrees. Now, when I say these things, please don't get perplexed and say that how am I going to, like accurately look at all these things. It just comes out of practice. Okay, so when you're just looking quickly at a pronuclei, your mind actually draws a line. Your mind draws a line through the polar body also, and then you imagine, okay, what should be the angle. Now, another thing which we have learned from time-lapse studies is, see, this alignment actually, can change, because this is a very dynamic process. So, initially it could be a parallel formation, but then before the nuclear membrane breaks, they can even orient it at the perpendicular form. Okay, so what happens is when you, at the time of checking pronuclei, when you observe something like this, when you observe a deviation from what you are expecting, the best way is not to discard it, but just to take it out and put it in a different droplet for culture. Okay, because if you have non-aligned pronuclei, if you have pronuclei present in the periphery of the oocyte, which is far away from the polar body, if you have unequal size pronuclei, see in this picture, you can see one is bigger and one is smaller. And these are all negative indicators for zygote quality. Okay, so it doesn't mean that we can't transfer this because live births have been reported, normal births have been reported. So the best thing is to kind of culture them in separate droplets. Now, the next feature which we can see is the nuclear precursor bodies, these tiny nucleoli which are present in the pronuclei. Okay, so you have to assess the number, size of this nuclear precursor bodies and the orientation. Okay, this is very simple to look at. So, mind you, there are few studies which actually say that these do not have any impact. But then there are a lot of studies which have actually correlated the quality of the zygote, quality of the embryo to the presence of this nuclear precursor bodies or the nucleoli. The reason being, these structures, they are the protein powerhouses of the cell. Okay, where all the proteins of any cell are constructed, and they are also the site of ribosomal RNA production. Now, how are they formed? So when the pronuclei, when they get closer to each other, the pronuclear chromatin differentiates and the nucleoli are formed. Now, what do we expect? Post-fertilization, pronuclei should contain 5 to 7 nuclei per cell. Okay, the emphasis here is on the alignment of this nucleoli in the equatorial region. So if you see something like this, the nucleoli aligned in the center of the pronuclei, then that actually indicates a normal formation. Now, as I said earlier, there are controversial results, but however, cytogenetic studies, lot of cytogenetic studies have suggested the association between nuclear precursor bodies and chromosomal disorders. Now, see, again, remember that the alignment of this nuclear precursor bodies is a dynamic event. So a single observation might be inappropriate and might be inefficient. Okay, but there are few features which actually, can be very important even with a single assessment. One, suppose if this nuclear precursor bodies are very small and they are scattered throughout the pronuclei, that actually indicates the early phase of pronuclear development. Whereas during the later phase, these small, tiny nuclear precursor bodies, they kind of merge or they kind of fuse and they become bigger, and which gets polarized at the junction. So that actually can give you an idea, suppose if you find these very tiny things, then you can expect a slow cleavage in those zygotes. And here, timing of observation is very crucial. And suppose if you are doing a lot of IVF, then here you can see the nuclear polarization would be much slower than that of the ICSI zygotes. Now, when you see nuclear precursor bodies, okay, so the preference actually should be given to zygotes, which has equal number and equal size nucleoli in both pronuclei. Okay, so if you have, say, for example, if they are aligned, they should be aligned in both. If they are scattered, they should be scattered in both. Okay, so if it's aligned in one and if it is scattered in another one, it is not given the first preference. Okay, so for example, something like this here, can you just see it here? Like here, the nuclear precursor bodies are kind of scattered, whereas here it is aligned. So it's not that we will discard this. You kind of keep it separately and culture them separately. Okay, when you have very pinpoint nucleoli, and that actually suggests delay in nuclear events, and it also indicates slow progression, and the embryo quality might be poor from this zygote. And suppose if the number of nucleoli are fewer than 3, okay, say for example, what you see here, you only see 2 here. Okay, this is also a negative indicator. Okay, and there also actually results in a high degree of abnormal chromosomal status. But mind you, I had told you this earlier, some of them can actually develop into good quality embryos and blastocysts and can result in normal live births. Hence, the key is culture them separately. I always insist in my lab that, segregation should happen at the PN check stage itself. Like all abnormal looking PN should be cultured separately, and the normal looking PNs should be cultured separately. And when I say abnormal means like these minor things. Anyway, like say, for example, 3PN and things like that, definitely you will not, utilize. But what I meant is like this nuclear precursor bodies, the changes, the deviations, whatever you see, you actually culture them separately. Now, the next feature is the cytoplasmic halo. Now, cytoplasmic halo, like you can see in this picture, there is a clear area around the periphery of the zygote. And this is also another visual marker of oocyte activation. See, normally when we don't see fertilization, right, like you can just see there will not be any halo. Okay, and that is an indication that oocyte activation has not taken place. Sometimes what happens is, so if you're doing the FERT check a bit late, okay, and then you have not seen the pronuclei, but then you see the halo. So there is some indication that, oocyte activation has taken place, and you can do a recheck around the 25 hours, 25 to 27 hours, and see if the first cleavage actually happens. Now, from where does this halo form? This is because of the withdrawal of cytoplasmic component from the oocyte cortex to the perinuclear region. And this is facilitated by the microtubules. Okay. And if there is presence of cytoplasmic halo, that actually is associated with the formation of good quality blastocysts. And these blastocysts also has an increased implantation potential. Now, again, observation time is very crucial. Then ICSI zygotes, you have, like it's more accurate because you know what time the sperm has been placed inside the oocyte. In IVF, we do not know. So this actually observation of cytoplasmic halo would be more accurate and more relevant in ICSI zygotes. Now, generally, we all do FERT check at 20x magnification, but cytoplasmic halo is best assessed at 10x magnification because that gives you a clear idea. Now, moving on to abnormal fertilization, like what are the abnormal features which actually you observe post-fertilization? First one is one pronuclei and one polar body. Okay, so this indicates that there is a parthenogenic activation of oocyte, that's an artificial activation without the contribution from the male gamete. Now, normally these zygotes have just one haploid set of female chromosomes. Another presentation is one pronuclei but two polar bodies. Suppose this happens following IVF, then this suggests that sperm penetration has happened and oocyte activation has happened, but there is failure of sperm head decondensation. Okay, but in ICSI cycles, it is suggested that oocyte activation has happened by the injection procedure. Mechanically, we have induced activation there. Okay, and you can actually observe this. Suppose in ICSI, you are seeing one pronuclei. You can just check after some time, maybe 12 to 24 hours later. That would help you to identify the oocyte with asynchronous pronuclear development, which actually might be a normal one, a diploid zygote. Okay, some cases, the single pronuclei, it could be from the fusion of two pronuclei, and there is evidence in the literature that these zygotes have been shown to result in term delivery. And it also could be due to clustering of some vacuolar or pronuclear membrane remnants. Now, there are a lot of healthy births have been reported from single pronuclei. And so what does this suggest? That whenever you see a single pronuclei with two polar bodies, do not discard it. They should be separated at the FERT check and they should be followed. And if normal cleavage occurs, you can consider them for embryo transfer if you do not have any embryo from 2PN zygotes, or, like if you have transferred blastocysts or cleavage stage embryos from the 2PN zygotes, and if it does not result in pregnancy, and if this is the only embryo which is left behind, you can consider transferring them. Now, if there is a choice, single pronuclear zygote with 2 polar bodies should be chosen over single pronuclear with 1 polar body. And because the reason is, when there is a second polar body which is extruded, that ensures that oocyte meiosis has been completed and the diploid character of the oocyte. And usually in ICSI, single pronuclear zygotes are less likely to be diploid, because in ICSI, you know, like we are actually injecting the sperm inside. And in spite of that, if it is a single pronuclear, the chances of them being diploid are less likely. Now, the next presentation is 3 pronuclei with 1 polar body. Okay, so I do not know how many of you actually look at polar bodies also during FERT check, because I see in many labs, they just only look at the PN. And unfortunately, in few labs, they don't even look at the PN. They think that PN check is not at all necessary, but I am a very strong advocate of doing fertilization check and then segregating at that stage. So this is one feature where, you know, when you see 3 pronuclei and a single polar body, this shows that oocyte activation has been normal, but the second polar body is not extruded. Instead, it is retained within the ooplasm, making the configuration abnormal with 2 haploid pronuclei from the oocyte origin and the 3rd haploid pronuclei from the sperm origin. Now, when this zygote leaves, it forms a triploid embryo, and in this case, it will be digenic triploidy, that is from the oocyte origin. And most of the 3 pronuclei arising from ICSI are of digenic origin, since only single sperm is injected. See, we assume that in ICSI that only single sperm is injected, but then I know, you know, a majority of the labs, especially you know, very experienced ICSI practitioners, they do not just take single sperm, they take multiple sperm in the pipette, and I know there is a fair chance of more than 1 sperm entering the ooplasm. You know, sometimes you are very skilled enough to aspirate the sperm back, but many a times what happens is you will not be able to do that, and this second sperm will also go inside the ooplasm. And that case, it is a totally different scenario. Your 3PN actually comes from the second sperm. Now, the next presentation is 3 or more pronuclei with 2 polar bodies. Okay, this again indicates oocyte activation is normal. And the chromosome configuration is that of polyspermy, because, you know, you have 1 haploid nuclei of the oocyte origin and the remaining nuclei could be of the sperm. Okay, it suggests diandry, and most of the IVF 3PNs are diandric in nature, like it is because of multiple sperm entering the egg. In ICSI, if you are very sure that you have taken only 1 single sperm, and if you have injected you know, like 1 single sperm, then 3PN actually could be because of cytoplasmic injury. It could be the fragmentation of nuclei or abnormal chromosomal constitution of the oocytes and sperm. Because sometimes, you know, the sperm, it could be diploid in nature, like it might have 2 set of chromosomes, and even though you have injected a single sperm, this chromosomal component will actually be triploid in nature there. Okay, but one feature which you will observe when there are this multiple pronuclei is the pronuclear size would be much, much smaller. Like more, in some cases, I'm sure you would even see 4, 5 pronuclei. And here they would all be very tiny, tiny like vacuoles. So this is one feature you will definitely observe. Now, this is, you know, like this information where when you are just checking your FERT check between 16 to 18 hours, the oocyte actually looks 2PN. But then when you are doing a early cleavage check, you know, then you will see that this 2PN zygote actually would have, you know, been, it will still stay as a single PN, or it would look like a 3PN. Okay, so this is actually, you know, this phenomenon is spotted much after the invention of time-lapse microscopy. But these embryos are shown to have lower implantation potential. Okay, when you see something like this, but normally if you do not have time lapse, you will not see this unless like you have the habit of doing an early cleave check. But if you see something like this, it is better you culture them, separately from the normal this thing. And see, once the cleavage happens, right, like it is very difficult. You cannot make out which one is from the normal zygote, which one is from the subnormal zygote. So try to culture them separately. Now, we know that like sperm, there are a lot of reasons. Motility can be a problem. Morphology can be a problem. DNA fragmentation can be a problem. These are the things which actually we can measure or we can just look at. But in the case of oocytes, right, like there are a lot of factors which have been identified for fertilization failure. Okay, I have given a list of possible oocyte-derived causes. See, some of them, it could be an aberrant GDF or BMP. This bone morphogenetic protein is a very important protein which is involved in fertilization. Now, if you have abnormal protein secretion, then what happens is the cumulus differentiation actually becomes abnormal. There is increased apoptosis. So, and this will help, will prevent sperm penetration or acrosome reaction. Similarly, when you have abnormal production or secretion of the zona pellucida protein ZP1, ZP2, ZP3, and that actually, result in altered zona pellucida structure, and the sperm cannot, undergo acrosome reaction. But in ICSI, it shouldn't matter because, like your acrosome reaction happens outside. But if it is IVF, then you can see it here. Similarly, there are a lot of things, like I do not want to take much time in going through all these things in details, but these are things which actually you cannot measure in a day-to-day practice. Say, for example, right, like ATP, GTP, or lamin B, all these protein deficiencies or increased secretion cannot be measured, but you can only, like attribute it. Like these also could be the reasons, because studies with fluorescent microscopy, studies with electron microscopy have shown us that all these factors play a very important role in the success of fertilization. Now, this is something which is really heartbreaking for us. That is the total fertilization failure. Okay, like when you were sure, say for example, you have a normal oocyte and you have a normal looking sperm, and the next day when you're doing FERT check, and then if you find them, completely unfertilized, and this is really heartbreaking. I know, like it is heartbreaking for the patients, but I know it is actually devastating for the embryologist. Many of them may not show it, but then inside you feel very terrible. You're always thinking that what is that I have done wrong, that, fertilization has not happened. But what is the incidence of this? Like some studies, they say 2 to 3%, but there are studies which say 1 to 5% of ICSI cases can actually result in total fertilization failure. And here they say that the inherent quality of the oocyte are largely responsible for regulating the mechanisms, okay, happening during fertilization. And we have to actually look at the oocytes here. We have to concentrate on the quality of the oocyte. And one of the key factor is oocyte maturation. Now, when we are talking about oocyte maturation, see, we can just look at two things under the microscope. One, we can see if the germinal vesicle has broken down. And then we can also see if the polar body is released. So these two things are there. And then we assume that the oocyte is mature. But there are a lot of other factors also, which is very, very important for the preparation of the oocyte for fertilization process. And like there should be a proper condensation and alignment of chromosome. The spindle actually should be formed and it should function properly. And there is fidelity of the chromosome separation. All these things cannot be visualized. Okay, so when you are not able to see a majority of the oocyte activities, and when you're just looking at two factors, it is not wise to say that I have given a mature oocyte. But fertilization has not happened, and that's something to do with the lab. Now, this picture, if you see, you can just see the nucleolar, the chromosome configuration here, the nucleolar configuration. You see the microtubules, you see the actin filaments. So all these things are actually important. There should be a proper relocation of these organelles. There should be synthesis of proteins, post-translational modifications of messenger RNAs. And all these things actually gets accumulated during oogenesis, and this is required for fertilization. So we are not able to see this. Now, like to a certain extent, nuclear maturation and the cytoplasmic maturation goes hand in hand. Okay, but not 100%. There are certain features in the cytoplasmic maturation process which is independent of nuclear maturation. So in case if that doesn't happen, then though the oocyte actually looks mature, meiotically, but then it is not actually ready for fertilization. And that is when you actually see low fertilization or total fertilization failure. Now, when we are talking about this oocyte maturation, remember, it happens during its stay in the follicular environment. Because the ovarian follicle, it actually provides an environment which will enhance, which will help in the growth of the oocyte development. A lot of that hormones are involved, growth factors are secreted, and this also helps in the regulation of oocyte meiosis. And then oocyte also, it derives a lot of nutrients from the granulosa cells and the cumulus cells, and there are a lot of molecule signals which happen. So oocyte maturation, it is just not only the factors which is directly acting on the female gamete, but it is also influenced by the regulators of the follicular environment. And when we are talking about assisted reproduction, to a great extent, this is subjected to the exogenous gonadotropins which are used in the ovarian stimulation. Now, all these studies, they tell us what influence an exogenous stimulation has on the ovary. Like oocyte metabolism is shown to be altered, ATP content is altered, transcription status changes, there is asynchronous nuclear and cytoplasmic maturation, microfilament distribution gets disturbed, and oocyte-derived embryo polyploidy happens because of, like the kind of stimulation, and the developmental competence of the oocyte will also get compromised. So, like all these factors are definitely involved in by the usage of gonadotropins during ovarian stimulation. Now, this is a very interesting study which was done as early as 1998, but there was also another one which was done in 2016 that in a previously failed fertilization cycle, they have made certain adjustments in the ovarian stimulation protocols, and they have shown an improvement in the fertilization. So basically what they have done is they have adjusted the time between the HCG trigger and oocyte retrieval, and they have also extended the timings between the cumulus, the denudation process, and this is shown to improve fertilization. Okay, and the concept here is you actually expose the cumulus oocytes to cumulus cells for a long period of time, and that would facilitate the acquisition of ooplasmic competence. Now, this is the paper. Like if you're interested, you can just go through this. There was one which is published in 2020 also. This one was published in 2016. Okay. So what they have done is 52 couple, they were confirmed negative for sperm-related oocyte activation. Okay, here they realized that it is not the problem of the sperm. The problem is with the oocyte. And what, like they were tailored superovulation protocol, and they found that significantly higher fertilization, like so 59% versus 2.1%. And in these cases, there was a clinical pregnancy of 28.6% versus 0%. But in both the cases, the maturation rate was found to be similar, meaning metaphase 2 oocytes were found. But then by altering that, they were able to improve the fertilization rates. Now see, if you just look at the timing between HCG trigger and oocyte retrieval, it's slightly higher when there is successful fertilization. Similarly, the timing between HCG trigger and the removal of the denudation process, here again, it is higher. And all of them actually, the longer the time, the better they got fertilization. But then what is interesting is, this is not something, like something, this is the normal one, like 29 hours is actually, like 34 to 36 hours, which is the normal one. But actually, in cases of failed fertilization, if the patient comes for the second cycle, you can think of slightly increasing it even more. That is what the take-home point from this particular slide. And then there is also another ovarian stimulation strategy, which was shown to improve fertilization resulting from immature ooplasm. And that is by adding a gonadotropin agonist to the HCG trigger. That is what we call it as a dual trigger. And there are 2 retrospective studies which reported that the dual trigger improved ICSI outcomes in patients with previous low fertilization rates. And this, they say, it does by enhancing the ooplasm maturity. Okay, these are the 2 articles which have said that. But I do not know, like these days, majority of the cycles are, I think, dual triggers. I'm not sure about what happens in the other clinics, but I know from our this thing that majority of them. So I think that portion has to be, is already taken care of. But what I tell our people is, when there is issue with fertilization, if the patient comes for the second cycle, we always allow them to do the denudation, closer to 40 hours and then ICSI at around 40 to 41 hours. This is, like an anecdotal recommendation. Now, the next one is, artificial oocyte activation. So this is when we think, or when we feel that there is a problem with the calcium release, okay, and which is found to be one of the attributable causes for ovarian activation, oocyte activation deficiency, then you can actually use artificial oocyte activation. There are 3 methods. First one is mechanical activation. I'm sure you would have read papers where, they say that aggressive injection, aggressive immobilization of sperm, as well as aggressive aspiration, where you aspirate the ooplasm couple of times and then put the sperm back during ICSI, that can actually help you in getting a better fertilization. Then there is also chemical activation, and we use a lot of this calcium ion channels and ionophores and things like that, which actually plays around with the calcium concentration inside the oocyte. And then you have electrical stimulation, where oocytes are subjected to electrical field. This is not something which is commonly used, okay? Here, this is the most common method of oocyte activation. And I know many of the labs do this also when they anticipate. But here the problem would be, suppose if the oocyte is immature, then any kind of aggressive injection, it would actually degenerate the oocyte. So this is kind of, like the embryologist has to be experienced enough to understand this, where, there is a chance of increased degeneration in the oocytes. But if you are not experiencing degeneration, then aspirating the ooplasm couple of times actually might help. But, there is no consensus regarding the efficiency, okay, in the improvement of fertilization and pregnancy rate following artificial oocyte activation. Sometimes they have reported an increased fertilization, but embryo growth is not good. In some cases, they've got fertilization and then they've also got embryo development, but there are no pregnancies. So, like we cannot say that for sure, okay, this will help. You can only try. Okay, and a paper which was published in 2015, this is a prospective study. It just showed that like calcium ionophore treatment successfully increases clinical pregnancy and live birth rates in low or failed fertilization. But then they have also suggested that instead of using one agent, a combination of 2 agents, an ionomycin and then, A23187, the combination of these actually can be used for increased fertilization. But with all these things, safety is a concern, because many of these oocytes, when ultrastructural study has been done, it has been found that there is a delay in the polar body extrusion. Okay, so now we do not know what are the downstream events. Okay, our aim is just to get fertilization, but it doesn't end there, right? Like there are a lot of downstream events which can have an impact on the epigenetic modifications in the zygote or the embryo. So safety is always a concern, though a very small study has reported that the percentage of abnormal or the babies which are born by this oocyte activation is much lesser. But I think we have to wait for a lot of data. And then there are also some studies which say that like if you use it in elderly people, your fertilization rate would be better. And then, like if you use it in cases where there are poor sperm parameters, your fertilization rate would be better. But personally, like from whatever I have read it from the literature, I would not attempt that unless it is warranted, because all these things can have an impact on the epigenetic modifications, and hence we have to use artificial oocyte activation very judiciously. So to sum it up, I think we have almost come to an hour of this class. So fertilization, it involves a series of consecutive steps that starts with the recognition and fusion of sperm and oocyte membrane to form a zygote. And these days, most of us, like do ICSI, and ICSI bypasses all these initial steps of natural fertilization, like penetration of cumulus cells, binding to the zona pellucida, and fusion with O lemma to get good pregnancy, good fertilization. Okay, first we have to kind of benchmark it. What is the international benchmark which is recommended? That in IVF, you have to get more than 75% fertilization rate. If a lab gets more than 60%, they are competent, but then you have to aim for more than 75%. In ICSI, again, it should be more than 75%. But, there are labs which actually routinely report more than 95% fertilization rate. I do not want to comment on that, but if I see a large number of cycles, okay, on an average, what we observe is the average fertilization rate is anywhere between 75 to 80%. And failed fertilization rate definitely should be less than 5%. And oocyte lysis rate in ICSI, it should be less than 5%. I think the skill has to be perfect. And it also depends upon the quality of the oocytes. If they are mature oocytes, then definitely your degeneration rate can be restricted to less than 5. But if it is less than 10, that the lab is competent. It doesn't mean that the lab is bad. There could be other factors which could be contributing to the oocyte lysis. Now, to achieve this kind of fertilization, it is, there are a lot of factors which could be responsible. It could be the female age, because in elderly patient, you have issues with the maturation. Then it could be something to do with the ovarian stimulation protocols. See, when I say ovarian stimulation protocol, what I mean is the response of a particular patient to the protocol. Because, see, the protocol which a clinician uses, right, it is something which is worked in hundreds of patients. And if it does not work in one patient, it just indicates that her response to that particular stimulation may not be right. And of course, semen parameters are also very important. Oocyte quality is important. And then last but not the least, operation process. The embryologist, has to be extremely skilled to get good fertilization. But more than all that, it's also important to understand the biology of fertilization, like what we have just gone through now, and then the various presentations, and kind of make assumptions based on the presentations. It is not necessary to by heart all these proteins, the growth factors, the cytokines which are involved in fertilizations like isomo-1, isomo-3 and all that thing. Those are not very relevant to clinical situation. But then we should actually know when you see a polar body, when you see 2 pronuclei or 3 pronuclei, and then you decipher so that, at least the subsequent cycle can be better. Thank you very much for all your time and for the patient hearing.
Thank you so much, ma'am. That was such a wonderful lecture. I would say it is very difficult for you to comprise this vast topic on fertilization, including physiology. As a clinician, Actually, it was a total recap of the entire textbook on fertilization. I really had a recap of physiology of fertilization first, and many insights which you said were very, very practical. For example, you gave insights on how to segregate abnormal looking PNs that are, in a separate droplet in the beginning, which is very important in the critical phase of fertilization. And more importantly, all the labs and all of us should have fertilization checks. That step should not be, avoided. And also, as a clinician, we feel that, we have given M2 oocytes, which you made, that really hit me hard, because we only know M2s. You said there are a lot of post-translation events which is not visible to the eyes that can have an effect on the fertilization. So I think that is a very important message. And also in previous fertilization failure cases where the sperm factor is totally negated, you gave us a very good insight on, stimulation protocol modification, or, probably delaying the denudation and, delaying the ICSI a little bit. Thank you so much for this wonderful lecture, ma'am. I can't really thank you enough from the bottom of my heart. And
Sure.
for all the benefits of all the delegates who are online and offline, we will share this recording because it is a wonderful lecture to keep hearing again and again in our clinical practice. I think Sindhuja will now do the final quiz, and then we will hand over the mic to Madam Ratna Madam.
I'm going to be launching the next quiz. This also has only 5 questions, so I encourage everyone to participate. Are you able to see?
Yes.
Okay. So all the answers are absolutely from ma'am's presentation. So if you've paid attention, I'm sure all of you will be able to answer. I'm very excited to look at the answers. So for me, it's like watching live election results. I can actually see how many are answering it right, like every question. So it's very interesting for me to see. It'll be open for 5 minutes. I'm going to try to share the QR code in the YouTube link as well. So for whoever is watching in YouTube can try. I'm not sure if it'll work. I really apologize if it doesn't, but I'm just trying. Trying. 75% have participated. I think we'll wait for another 1 minute. Okay. I think 80% have participated and it's almost 5 minutes. So I'll end the poll.
Yes.
Yes, please.
Yes, I'm going to share the results. I think the current answers are visible to everyone.
Yes, that is visible.
Yes. So to summarize, the first event to be assessed microscopically after fertilization is, so the correct answer is by 73% of the people, it's the extrusion of the second polar body. So that's what we can assess microscopically. So cytoplasmic halo is a marker of oocyte activation. Excellent, 83% have answered it right. Next is I thought it's a tricky question, but everyone answered easily. So all of the following are abnormal fertilization except, so the normal fertilization is 2P and 2PB, which 68% have answered. And the percentage of total fertilization failure in ICSI is absolutely right, 2 to 3%. So 76% have answered it right. The benchmark of ICSI fertilization rate is more than 75%. 75% have answered it right. So I think Sujata ma'am is very happy.
Thank you so much, all participants. We have few questions on the chat box. Sindhu, if you can just take that.
Ma'am, can we hear from Ratna ma'am?
Yeah. Ratna ma'am, good evening. We would like to have
Good
your
evening.
short
Yes.
insights and concluding remarks before we get
Yes.
to Q&A.
Thank you, Sujata, for
Thank
such
you,
an
ma'am.
outstanding presentation.
Thank
You are not
ma'am.
only a good orator, but also a good teacher.
Thank you, ma'am.
And that the quiz is the reflection of that. And you described this complicated process thoroughly but precisely. Thank you. And yes, got some more information from you also.
Thank you very
Thank
much,
you.
ma'am.
Thank you. Thank you.
Thank you, ma'am.
Thank you so
Thank
much.
you, ma'am. Shall we proceed
Yes.
to the questions in the chat box, ma'am?
Sujata ma'am, 2 minutes
Yes.
for two
Yeah,
three
yeah.
questions which is there.
Yes, yes, sure.
How can we differentiate smooth endoplasmic reticulum or inclusion body?
Okay. See, smooth endoplasmic reticulum actually looks like a flattened disc. That's, like if there's a picture, it would be much, much easier. But then, it would, it is like a large disc and kind of flattened and smooth it looks. But this inclusion bodies are tiny objects which are more dense in appearance. Definitely you can differentiate only. I'm not sure if I have a picture of that. Let me just see.
Okay, ma'am. While you're looking, I'll just go to the next question.
Yeah.
Ma'am,
Yeah.
which embryo is good in cleavage stage, equal or unequal cell for transfer? So I think we all agree equal cells of blastomeres is definitely more ideal.
Yes.
And Vishal has asked a question. It is quite a big question. I answered to him separately. So I said it could look like an empty follicle syndrome. or
I think it is
or you know, a trigger
it is a master
problem.
class in itself. He has opened a topic for another master class.
Correct.
I think we can
Yes.
answer
Yes.
this.
Yes. So
Yeah.
I've told him that it could be a case of empty follicle syndrome or receptor polymorphism. And there's another interesting question from Rajesh. He's asked, what is the role of sperm centriole in fertilization and early embryonic development? I think ma'am touched upon that. It's a very good question. Ma'am, you want to add a point?
No, centriole actually, that is the one which actually helps in the formation of the spindle, the alignment and all that, like where the pronuclei gets aligned, right? Like centrosome, actually, the pronuclei is brought together to close to each other. It's because of the centrosome, the centriole.
Yes.
Yes,
So
actually,
that's why
it forms,
when, yes,
yeah,
yes.
it forms the MTOC, the
Yeah.
microtubule originating centriole. And
Yes.
it attracts the two pronuclei to come together, rightly said Sujata.
Yes, see this,
MTOC, formation of MTOC,
yes.
microtubule
If you can see this picture, right? Like, see, can you
Yes.
see something? This is an inclusion body, like, but whereas if I do not have the picture of smooth endoplasmic reticulum,
Yes.
smooth endoplasmic reticulum would look much bigger and then it would not look this dense.
Yes.
It would be a
Dense,
very
yes,
thin
yes.
disc.
Yeah, the flattened disc,
Yes.
rightly you said.
Yes,
And
yes.
you know that smooth endoplasmic reticulum, it may be just cause of fertilization failure even after ICSI.
Yes.
And inclusion body, sometimes it looks a refractile body. It is
Yes.
very dense and
Yes.
bright.
Yes.
It
Yes.
looks like
Yes.
under microscope.
Yes.
Mostly everyone's appreciated, saying it was a wonderful presentation, and thanking you, ma'am. Just the
Thanks, my
last few
pleasure.
questions. What do you think about being very mucus cumulus during denudation? I think they're trying to ask very sticky cumulus during denudation.
Yeah, see, the thing
Probably
is,
it's association with
like,
fertilization they want.
yeah, see, what happens is when you find such cumulus, right? Like it is, and when you aggressively try to denude it, you will end up with a lot of degeneration. Because, like, say, and I feel that to a certain extent, you can increase the exposure to hyaluronidase rather than putting it through mechanical denudation. These are features which actually could be innate. It has nothing to do with what is happening in the lab. So here, when you find that the first one is very sticky and it is not getting if you are not able to denude it properly, then you just leave it in hyaluronidase for an extra 10 to 20 seconds. And then you try to take it out, but don't aggressively mechanically denude it, because like there is a lot of movement of polar body which can happen in the perivitline space, and then you could be directly injecting your sperm into the spindle, meiotic spindle, or sometimes what happens, you also can cause degeneration.
Yes, actually maybe a immature oocyte sometimes, or maybe reflection of the improper expansion of the cumulus due to some defect in the metabolism.
Yes.
Hyaluronic acid is not properly released, that expansion is not properly done.
Thank you, Varsha, for sharing a picture of an SER. Very thoughtful of you in the chat box. And there's another question. What do you think about rapid cleaving embryos?
Okay, see, sometimes when like rapid cleaving embryos, it can be because of so many reasons. So, for example, if an 3PN zygote, it would cleave very rapidly. So it could be because of any ploidy reasons. But then culture conditions or culture media can also have an impact, because I remember when we were trialing a particular single step media, we would see morula on day 3. I'm not even talking about compacting, where we would just get properly formed morula. And then we had conveyed this information to the company, and they had tweaked some components there in the culture media. And so where we started to see compacting on day 3 morning, day 3 afternoon, they would be completely compacted. So, like your media components also can play a role. And say these are the two things, but other than that, it could be an oocyte phenomenon as well.
Yes. Vitro life, usually with vitro life, the cleavage is much earlier than the other. Yes. And sometimes maybe chromosomal abnormality also.
Yes, ma'am.
Yes, ma'am. I think we'll take one last question, ma'am, because I think we're all
It's
past
already 9
dinner
o'clock, and then I
time.
think the
I'm really
questions will be
sorry about
endless
this.
because, yeah, sure.
Yes, ma'am. Last question, ma'am. Abnormal fertilization and influence on genetic screening results. Because I
Yeah.
think that will highlight, because there's another question on how important it is to do PN check. So I think this could answer both.
Yes, already.
Yeah, see, the thing is, like suppose if you have, like 2 PN, which is actually changed to 3 PN, and then you get a good out of it, a good blastocyst, and you can subject this for genetic analysis, then you will know for sure what is the chromosomal status. So when you see, or even if it's a single PN, okay, but then you got an excellent blastocyst. So if there is a facility to do biopsy and then send it for genetic analysis, you can confirm the ploidy status rather than just taking a chance on it. Earlier, they didn't have. So they just went ahead and transferred. Now that you have the facility for pre-implantation genetic testing, I think it is advisable, especially when you see abnormal presentations, to confirm.
Ma'am, one last question from my side, if I'm permitted to ask, is like oocyte and sperm factors and all this in affecting fertilization, just key points which can affect fertilization in the lab environment, something that we need to take care and take home.
Okay, so I will just give you an incident which happened in one of our labs in much earlier days. Okay, we had a benchtop incubator, and this benchtop incubator was serviced, and we started to use it. But unfortunately, like we had a huge batch and we had to use it. And what happened was we cultured, okay, some 3 patients' embryos in that particular benchtop incubator. And the next day, we found there was complete fertilization failure in all of them, all the cases. Okay, and then when we just looked at the pH, okay, which since what happened was we didn't have a pH meter, we borrowed it from one of the vendors, and then when we checked the pH in the culture dish, we realized that the pH was more than 8, more than 9. Okay, so we realized that there was no CO2. The CO2 was flowing, okay, we could actually see it, but then it was not flowing into the chamber. So, like carbon dioxide levels can have an impact on your fertilization. Then your temperature can have a major impact, when suppose if the incubator gets turned off or your incubator is not calibrated properly, then that can also have an impact. See, especially in the lab, like you cannot pinpoint on one thing. Every single thing can be a reason. Like it could be just your pickup this thing where your oocyte gets exposed to the ambient conditions for a very long period of time. Or it just could be the dish preparation where, like you make droplets and it is not covered with oil immediately. You make 3-4 dishes and then try to cover it with oil. That can actually compromise on the osmolarity and that can have an impact. So like this, every small thing can have an impact. Now, when does this impact become obvious? When the quality of the gamete is compromised, it becomes obvious. If it's a very robust, like an young patient, 26-year-old patient, okay, and then you put it in the incubator, power goes off in the night, 3-4 hours, there is no power, okay, and in spite of that, you get embryos, and in spite of that, you get pregnancy. Does it mean that if you turn your incubator for 3 hours in the night, all of them will go through this? No. Here, it depends. If the gamete quality is bad, even a very small insult can have a major impact. So in the lab, if you ask me, every single thing is important.
Thank you so much, ma'am. Thank you.
Yeah.
That's Vasundhara ma'am's line too, ma'am. You both have the same lines. That's what ma'am says too. Everything matters.
Yes.
That's
There's
right.
only one thing that matters, everything.
Yes.
Yeah. Thank you so much, ma'am. At
My
the
pleasure.
outset, I would like to extend my heartfelt thanks to Sujata ma'am and Ratna ma'am for spending the entire evening with us, dedication and teaching for all of us. It was really a wonderful lecture, a wonderful teaching for all of us. Thank you so much for your time, and thank you all the delegates for spending your time in this, education platform. We would be doing this on a monthly once basis, and next month we would be having a webinar on all about implantation, and Dr. Sandeep Karunakaran will be talking and Dr. Swaminathan will be chairing the session. We will be sharing all the details of the next upcoming webinar and also share the recording of this wonderful A to Z of fertilization recording with all the delegates. We request you to kindly post your feedback, which we have shared the link in the chat box, which will help us improvise in future. And since we exceeded the limit of 300, we were not able to, make amendments to this, and we shared the YouTube link. We would look into it the next time and also try to rectify that factor also. Thank you for bearing with us in this, small glitch that happened. Thank you so much, ma'am. Thank you so much. Thanks, Sindhuja, for
Thank you.
taking
Thank
your
you.
time and effort and, making this seamless. I really appreciate all your efforts. Thank you so much, Sindhuja.
Thank you. Thank you.
Thank you,
you
ma'am.
all.
Looking forward to
Bye-bye.
interact with all of
Bye-bye.
you.
Thank you.
Thank you,
Thank
ma'am.
you.
Thank
Bye-bye.
you. Good night.
Bye.
Good night.
Bye.
Good night.
Good night.
Good night, Srilata.
Good night, ma'am.